I was very glad at the end of session 1 to hear a pediatric question come up, and that basically made me feel good. I'm Gil Burckart. I'm Associate Director for Pediatrics in the Office of Clinical Pharmacology at FDA. And so I was being, pediatric oriented. I was very glad to hear that come up. That obviously means that people are thinking about that in relation to what we've discussed this morning. And my first slide here really is to kind of set the stage for our discussion this afternoon. We have 3 15-minute presentations and then our discussion. And basically it's because, that asks the question, why is modeling of BA and BE important for drug development in pediatrics? And so it's important for you to know, if you don't already, that BE studies on new drugs are only conducted in healthy adult subjects due to ethical considerations. So from an ethics standpoint, we can't do those studies in pediatric patients. We have to do them in healthy adults. And also, generic drug formulations approved for pediatric patients are not tested in pediatric patients. And therefore, the effect of ontogeny, all those things we've talked about this morning, are not considered. The effect of excipients and solvents may be very different in pediatric patients, but we don't test them. And the concurrent administration of other drugs or the food effect, which we were just discussing, may be very different in pediatric patients, but we haven't considered that. So those are the topics that our speakers this afternoon will be talking about. We'll have a panel discussion, and then there is a roundtable later. But we're going to have 3 presentations, one from Dr. Lucy Fang at FDA. We're going to have Dr. Liu from Bristol Myers Squibb and Dr. Sagawa from Pfizer. And so we'll have 3 15-minute presentations, and then we'll have a panel discussion, and we'll briefly introduce our panel members at that time. So let's go ahead and, let's see. These are the 3 speakers that we're going to have, and you have the biographies on the table if you'd like to look at the backgrounds for our speakers, and we have a fairly large panel and a panel discussion afterwards. So let's go ahead and get started with Dr. Fang.
So hi, good afternoon. My name is Lucy Fang, and I want to thank the organizing committee for inviting me to share with you on the regulatory applications and research of absorption modeling for pediatric products. So this is my disclaimer. The views expressed in these presentations are mine and do not reflect the official policy of the FDA, and the case discussed are for illustrative purpose only. So I want to first introduce the 2 concept terms. The relative bioavailability is used in the context of new drugs, such as comparing the pediatric and adult formulations, because the new drugs can potentially have greater changes in the formulation. So the BE limits do not necessarily have to be demonstrated. The sponsor can either come in to justify that the difference in the rate and the extent of absorption do not affect the efficacy and safety in pediatrics, or the sponsor can come in and to propose a different dosage in pediatrics. The bioequivalence is used in the context of generics. It's used for the determination that the generic product is suitable for substitution in the pediatric patients. Therefore, the BE limits need to be demonstrated. In terms of relative bioavailability, the guidelines from different regulatory agencies recommend the study to be conducted in adults. I won't bring your attention to the EMA guideline. It states that the study is preferred to be conducted in the adults, but the applicant should justify that study results can be extrapolated to the pediatric population. So before I go into the generic product with pediatric indication, I want to emphasize that approved generic products are considered therapeutic equivalent in pediatrics. Generic products are considered therapeutic equivalent and suitable for substitution if they are pharmaceutically equivalent and bioequivalent. Generic products are expected to have the same efficacy and safety profile when administered to patients under the conditions specified in the product labeling that includes all indications and all patient population, including the pediatric population. In a review of 12 years BE data, the results showed that the average difference in Cmax and AUC between the innovator and generic products are within 5%. So the PK difference between the generic innovator products is really small for generic products to be approved in the US. For establishing bioequivalence for pediatric generics, FDA generally recommends that BE studies be conducted in healthy adult subjects, and the BE outcome in healthy subjects can be extrapolated to the pediatric population. So this is consistent with the ACHE 11 guideline, and healthy subjects are considered the most sensitive population to detect the formulation difference. And the BE outcome in healthy subjects have been used to support the use in all populations, such as the patients with renal impairment or hepatic impairment. The same reasoning can be applied for the pediatric population, unless there is a concern of impact of age on the drug availability due to different formulations. So in the most recently updated 2021 BE guidance for the ANDAs, the guidance states that if the drug product is intended for use in the pediatric patients younger than 6 years, the applicant should justify that BE study results obtained from the adult subjects are relevant to the pediatric population. FDA recommends that the justification include formulations supporting the inactive ingredient in the proposed product are appropriate for use in the pediatric population. So this is consistent with the ACH guideline, the EMA guideline that I just went over. So when we extrapolate the relative bioavailability and BE outcome from adults to pediatric population, we have the following considerations. First, what is our degree of certainty that differences in absorption of different formulations in pediatric patients are adequately detected in adult volunteers? Second, how do we identify products where we should be cautious? Third, what will be our approach if high-risk products are identified? First, let's look at the use of PBPK model in the new drug development. FDA's Office of Clinical Pharmacology summarized the application of PBPK in the record submissions from year 2008 to 2017, and also discussed its impact in the product labeling. As you can see that the pediatric PBPK modeling in the NDA submissions are relatively constant, about 15% of all the submissions containing PBPK modeling. And the main intended use of those pediatric PBPK modeling is to predict PK and propose those initial dosage selection for pediatric trials. It has been argued that the pediatric PBPK modeling is preferred in kids who are younger than 2 years old over the allometric screening for drugs that the ontology is important for the drug's ADME properties. There are also cases that use pediatric PBPK modeling to evaluate the relative bioavailability prior to pediatric trial initiation. And also pediatric PBPK modeling has been used to simulate the untested scenario, such as drug-drug interaction in the pediatric population. So now let's look at the food effect. Food effect studies are conducted in healthy adults to characterize the drug and food interaction that may affect the efficacy and safety. However, we all know there is a substantial difference between the adults and pediatric in terms of the physiology and the food impact that may cause a different food effect in the pediatrics. Dr. Burckart and his group conducted research to really identify oral drug products that has food effect observed in adults and to really assess that implication in the pediatric population. So out of the 102 oral drug products approved for use in patients younger than 6 years, 43 products recommend the consideration of food intake in the drug labeling. And out of that 43 products, and 65% recommends taking the drug with food or with very specific instructions. And 100% of those taken without food were approved also in the pediatric younger than 2 years of age. So those aspects can really help us identify those oral products that have significant food effect in adults, but also need additional monitoring or assessment in the pediatric population. So additional methods such as the pediatric BCS classification system and pediatric PBPK modeling are needed to help us accurately assess the pediatric food effect. So for generics, we initiated this project back in 2017 to address the question, is bioequivalence established in adults relevant for pediatrics? We had a session and also roundtable discussion in that year's AAPS meeting. FDA conducted some proactive research projects. In the first project, the generic drug substitution in special populations, we collected clinical data on approved generic drug substitution in the pediatric population to really help assess, is there a signal? Is there increased risk for the generic drug substitution in the pediatric population? And our results really didn't identify any of this signal. And in the subsequent research contract, risk mitigation in the evaluation of relative bioavailability of pediatric generic products with University of Birmingham, we developed the risk assessment and mitigation tools based on the BCS classification system, the biorelevant in vitro dissolution testing, as well as the PBPK modeling. So we first developed a database in which the relative bioavailability studies as well as the B studies were conducted in the pediatric patients as part of the pediatric efficacy and safety trials. We really want to identify the risk factors associated with the drug substance as well as the drug formulation that can contribute to the failed BE or different relative bioavailability in the pediatric population. And the most notable risk factor is the physiological factors that may affect the ADME property of the drug. And for example, the age-related change in the absorption in terms of the age-related GI mobility, GI fluid volume or composition, and GI transit time. And we also noticed a higher intra or inter-subject variability in those pediatric trials that may contribute to a different outcome in the pediatric trials. And also I want to bring your attention on the poor study design. For example, that the really limited small sample size that may also cause to a different outcome in the pediatric trial. So from the aspect of risk assessment to identify the scenarios where the adult BE study may not reflect what's happening in the pediatric population, we really need a better understanding of the developmental changes across the broad pediatric age group. For example, that the age-related physiological change, as I mentioned in the previous slide. In addition to those, there are some other factors such as the drug substance or formulation characteristics can also play a role. So PBPK modeling can really serve as a tool to evaluate the interplay between the population-related factors and the formulation-related factors. We really believe that advancement or enhancement in the PBPK modeling can help us identify the underlying mechanism that can contribute to the different BE outcome or different relative bioavailability in pediatric, if there is any, and also can help us assess the predictive performance of the adult BE studies to the pediatric population. And in this research, we also explore the biorelevant dissolution testing because there is known variability in the GI fluids as well as the difference in the fluid volume in children compared to the adults. So use carbamazepine as a model drug. We first evaluate the impact of in vitro dissolution medium volume as well as composition on the carbamazepine dissolution, and we found that carbamazepine in vitro dissolution are not so sensitive to the composition of the biorelevant dissolution media, but the carbamazepine in vitro dissolution is sensitive to the volume of the in vitro dissolution media. And then we integrated the biorelevant in vitro dissolution profile into the PBPK model to predict the PK and PD outcome in adults and the pediatric population. And those virtual bioequivalent stimulation really shows that the product is bioequivalent in both adult and the pediatric population. So this project really demonstrated that integrating the biorelevant pediatric dissolution profile into the PBPK model can help us predict the in vivo PK and the outcome of generic drugs in the pediatric population. So some of the take-home message of my talk: the absorption modeling can incorporate the characteristics of the drug substance and formulation, as well as the pediatric physiology, to assess the potential differences in absorption of different formulations in pediatric patients for both new and generic drugs. Little data, if any, are available showing that the bioequivalent generic products in adults are inequivalent in other populations, including the pediatrics. The 2021B guidance for ENDAS recommends the applicants conduct comprehensive formulation comparison, in vitro characterization, as well as modeling simulation analysis as risk assessment. So finally, I want to acknowledge my FDA colleagues as well as the external collaborators for their contribution to the research in this field. Thank you all for your time.
Thank you, Dr. Fang. I wanted to mention for those of you that are online that Dr. Rebeka Jereb is taking notes of any questions that are in chat, and we'll try to get to some of them during the panel session. The next presentation will be by Dr. Liu of Bristol Myers Squibb.
Thank you very much. Good afternoon, everyone, and good morning or good evening in the world, in the online. Before I start, we have the title on the screen. Before I start, may I just do a quick survey in the room? How many people have answered yes to this question? Raise your hands. Or no? Is pediatric absorption model ready for BE assessment? You think it's yes? Nobody say yes. That's good. Okay. Yeah, we have one yes. Okay, let's move. So I have 3 observations as the end user of Gash Plus and Siva. I'm working in BMS Bristol Myers Squibb. As general, all what I share here is only represent myself. My own opinions doesn't reflect what the company is doing. And I have been working in Sims for 7 years, and I used Gash Plus for a couple of years as well. I'm regularly using Simseed. I may be biased, but as I shown on this slide, PBPK is big giants. What I'm sharing is a really piece of this big giants. I may be wrong. Let's move forward. So before touching the details, a quick remind, our gut is not only for the absorption, it also contributes to the disposition, metabolism, and excretion. For the oral drug, as you can see on the right gray area, we have the stomach, we have the intestine, and also colon, all those components, and come back to through the passive processes and also active processes, the drug gets into the central compartment. Usually, when we talk about the IV PK profile, when the drug gives as the IV in the morning, in lots of the examples people have shown, we always using the IV profile to define the distribution volume and also define the IV clearance. Sometimes people use the reticulate model to get the hepatic clearance. So here, mostly I want to emphasize the gut contributes to the disposition and the metabolism, and do not underestimate the contribution from the gut when we analyze the IV profile. So my 3 observations I describe in 3 questions, and really try to using a new terminology about the anatomy. It's about the assumption, not the absorption. It's about the data, not the disposition. And also it's about the right model, and try to answer if we have the right expression for the PBK model. By the way, we have been talking about the local exposure or the locally acting drugs, and also in the pediatric, especially we are interested to look at the drug disposition in the laminar propria. Either the drug can be orally, can be intravenously given. How we can get the best prediction of the drug exposure in the laminar, that will be our target. So here, my first question about the right assumption. Left-hand side is a traditional pH partition hypothesis. 2 keywords here. One is the free concentration gradients for this pH partition hypothesis. Another keyword is the bidirectional. Between this membrane, depending on the concentration gradients, and all this concentration must be free concentration, unbound, unanalyzed concentration gradients to define your drug absorption or the passive permeation. And also it's bidirectional. Now look at the right-hand side, I summarize the model I have been using, either ACAT or SimSip, Adam, or the Madam model, depending on which model you are using. As I said, could be based on the single condition in SimSip using Adam model, can be one directional, only from the lumen to the system. This is different from the left-hand side, the pH partition hypothesis. Assumption can be made, but think about that. And then we can see ACAT model on the top is using the total luminal concentration and the free enterocyte concentration to define the drug absorption rate. And it's not a single condition assumption, so it's bidirectional. But here again, I emphasize is using the total luminal concentration and compare with the free enterocyte concentration. Maybe it looks very, very detailed scientific, technically, but this is fundamental of the model absorption compared to the pH partition hypothesis I want to emphasize. The last one I'll share here is the Madam model, more mechanistic Adam model. Here we can see is really consistent with the pH partition hypothesis based on the free concentration gradients and also using the bidirectional absorption. So you have the lots of experience. See, I have using GashPass, have been using SimSip. I have got a good experience, have seen the success. To me, which is the right model we should use, and then how are they performing different? I was using really a simple case to demonstrate they really perform significantly different. So this is a theoretical model. Assume we have the gut, we have the plasma, they interact with each other. If you based on the free concentration hypothesis, then these 2 compartments eventually should get the equivalent status. So by changing the FU unmodified in the gut or changing the permeability from the plasma to the lumen, we can mimic what has been done in GashPass or what has been done in Adam model or in Madam model. If using the Madam model, that's the model D, purely based on the pH partition hypothesis, at the beginning, the drug is 100% in the gut. After a while, at the end, you will have the equivalent, and then your intracellular, your plasma concentration and your luminal concentration will be equivalent. That's the ideal case. And in this case, we will not see more than 50% of drug will get into the system. But if you are using model B and D, which is unidirectional absorption, are either using the free concentration in the lumen or using the total concentration in the lumen to drive the one direction absorption, your drug will be nearly 100% get into the blood. That's based on the Adam assumptions. And then you will say, oh, my drug is a high permeability drug. And if you are using the first model, basically using the total luminal concentration and the free plasma concentration, now here you will not get the equivalent at all. Maybe I have used quite a lot of time for the first case questions, and my next two questions will be very, very simple. Here is about the data. So we are talking about the pediatric absorption model. And at the beginning of the year, from China regulatory officer, they have guidance released, and using the PBK model to support the pediatric population formulation development is really encouraging us to try using the PBK. I was cautious to read the document, and the last I highlighted paragraph is one sentence, say in case of the sufficient data, unnecessary clinical study can be waived. Now, do we have enough data? Or more important, do we have the right data to drive or parameterize our pediatric absorption model? So in our team, we have been spending quite a lot of time and resource to collect the data to parameterize our PBK model. As I shown on this summary, we have quite a lot of data for the Caucasian, Japanese, and Chinese in fasted or fed state. Also consider the age dependency. But the more important, you will see lots of the books actually is blank, have nothing there, and is not available for us to collect from literature. Even important, I want to highlight through this exercise is we haven't really found a single study, well-controlled study, compare Chinese pediatric, Caucasian pediatric, and the Japanese pediatric in one study, head-to-head. All the data we collect on this literature is separate observed data. Maybe here is a call for the society to work together as an international group to do such a study. And we in the morning look at the local acting drugs in the pediatric. As I mentioned at the beginning, we are interested to look at the laminar peripheral concentration using the PBK. So left-hand side is GASH plus ACAT model and also PBK. Right-hand side is the SIMSIP whole body PBK with the MAD model. As I highlighted in two questions on the bottom of the slide, really for the GASH plus ACAT model, we kind of find the laminar peripheral compartment in the ACAT model. Even for those GASH plus users, you definitely know better than me. In GASH plus, they have lumped the gut to the rest of the body, that compartment. And for the SIMSIP MAD model, if you are a SIMSIP user, you know SIMSIP has a adult simulator and also pediatric simulator. Actually, MAD model currently not available in the pediatric simulator. My last two slides actually is giving you an example to highlight it, the MAD model, the functionality or beauty of this model. Here is a self-feeding. Self-feeding is a kind of similar to the caffeine. Purely is passive with high permeability, amount fraction in plasma nearly 50–60%, and no transporter in this drug disposition. So for this one, if we give the IV and the half-life roughly 7 hours, and if you give the activated choco, now you detox the self-feeding, and the half-life actually will be half. I tried very hard. I tried to do that in Adam. It's not possible. I tried also do that in ACAT model. Unfortunately, I don't know what's the best way to do it. But in simple matter model, actually using the exhibition effect is very, very simple, very, very easy, can predict the peak profile with a activated choke. And also with the matter model, now we can see how drug actually squeeze out from the system back to the lumen and get reabsorbed if without the activated choke. Now with activated choke, now the drug will not get reabsorbed, actually have more drug leak out from the system back to the lumen, and nearly 45% of your drug is in the activated choke, and eventually this drug will get into the feces. So as you may have imaged, so my answer to my question at the beginning to my talk is already there. I want to bring back the first slide I put here, is really gut is not only for the absorption, and we need to consider that in the disposition and also for the excretion, especially when your drug is a PGP substrate. Do not think about this IV clearance after the IV purifier is only the hepatic or the renal. You will have the antihepatic excretion, you may have the enteric enterocyte excretion, or even direct intestinal secretion contribute to the metabolism. So without the mechanism or absorption model, unless your IV profile, maybe need a bit more cautious. And this is my last slide, take-home message. I have one message. Think about your model and how you confident use your model. Are you the right modeler, or we have the right model and have made the right assumption with the right data to parameterize our model? That's all. Thank you very much for your listening.
Thank you very much, Dr. Liu. Our final speaker before we have a panel discussion is going to be Dr. Sagawa from Pfizer. Dr. Sagawa.
Well, thank you. My name is Kazuko Sagawa. I have been conducting the PBPK modeling, mainly focusing on absorption modeling for different dosage forms at Pfizer. Today I will talk about our recent experience conducting the virtual bioequivalence trial using the PBPK modeling for pediatric formulations. Before I go on, I would like to acknowledge the team that worked on this project. This is the collaboration between pharmaceutical sciences, PDM, Clean Farm, pharmacometrics, statistics, analytical. All those colleagues at Pfizer worked on this project for many years. So the case example that I will talk about today is a tofacitinib. I will just go over the dosage form, history of the dosage form development, and also talk about how we built the PBPK over those years using those different formulations, mainly in adults. I want to focus on how we conducted the virtual bioequivalence trial using the PBPK modeling, well, then our regulatory experiences. And lastly, I will just briefly touch on pediatric extrapolation, but I won't have the time to talk about how we actually modeled the pediatric in the PBPK model. So tofacitinib is a JAK inhibitor approved for the treatment for the rheumatoid arthritis. This was launched first as IL tablets dosed twice daily. And years later, several years later, we launched the once-a-day dosage form. This is the modified release dosage form. And these two formulations were bridged with the clinical bioequivalence trial in adults. And so several years later, we developed a pediatric oral solution dosage form. And this is equivalent to the adult IL tablets. And these two formulations were bridged with the clinical bioequivalence trial in adult healthy volunteers. So now we are developed and fighting for the pediatric once-a-day dosage form, and this is another modified release dosage form, and these are the beads that conveniently be able to administer for young children. So this formulation needs to be bioequivalent to the pediatric oral solution. So the virtual bioequivalent trial that we are conducting using the PBPK is to bridge those two pediatric formulations, oral solution, to once-a-daily pediatric modified release beads. So first, we developed the PBPK model to just fix the systemic component. So first, we used the first-order absorption, used the IV data and multiple DDI clinical study results to make sure that our enzyme kinetics is very well established for this product. That part was important to be able to extrapolate to the pediatric using the ontogeny that we can use as an ontogeny as a function to extrapolate to the pediatric population. So we made sure the enzyme kinetics and also flu PBPK is incorporated into the tofacitinib systemic component. Then we incorporate the absorption component with measured experimentally determine the PKM properties, solubility data, and so on for IL products. Tofacitinib is a highly soluble drug, so for that matter, absorption part for the IL component was relatively simple. So then we moved on to the ML dosage form simulations. Then first we developed the model for this adult ML dosage form that I mentioned, and those are the osmotic delivery system, where we have a very good confidence many experiences using the in vitro dissolution data to predict the in vivo drug release. And this is where we had confidence in using the in vitro dissolution data to model the adult once-a-day dosage form ML formulation. So then, and that was used to make sure the permeation setting for the lower GI is appropriate for the tofacitinib. Or then we used the beads formulation and tested in adult healthy volunteers to make sure the scattering of the beads, the GI transit of the beads, they are appropriately incorporated into the model. And there are many literature data to explain the scattering of the beads in the GI tract, but that is really depending on the size of the beads, density of the beads. So our beads are relatively smaller, lighter in density, so that not necessarily we can incorporate those information from reported literature data. So that was used for the first set of data. So the bar graph shows so many multiple studies that we used to verify this model, and made sure that there are study-to-study variability, obviously. So this is a plot for the predicted versus observed ratio. So the one obviously means that we are completely predicting. So we made sure the model is not biased towards either underestimation or overestimation. Just to talk about this complexity of the modeling for this MLBs for the pediatric population, this was the enteric-coated bees, and this was the modified release enteric-coated bees, and we had multiple clinical studies tested for the different durations and different coating levels. So we utilized those information to build the PBPK model to reflect on how this bees is released with a different pH and different the duration of the release. As it was discussed earlier today, there's no single in vitro dissolution data that actually capture how it's released, ever changing the pHs and fluid volume in the GI tract. So that was, unless the tool gives you the option to use the enteric-coated beads, which was not the case at that time, well, still now. So we mathematically treated the in vitro dissolution data measured at the pH 6.8 to reflect on how there's a gap because it doesn't release in the stomach, but it will release in the small intestine. So that was mathematically treated to modify the measured in vitro dissolution data to reflect on that. So I think this highlights still the limitation of using the just a single in vitro experiment dissolution data to directly incorporate into the model, if especially the dosage form is very complex. So then we conducted the virtual bioequivalence trial. Then the first thing we did was a sample size analysis, and this method is generally used for any clinical trials. So instead of having a clinical trial, we generated the 5,000 individual PK profiles for the reference formation, which is the IR oral solution. Then we assumed a 10% geometric mean ratio and incorporated the inter-subject variability, which was estimated from the clinical trials. So this is the part that it gets a little bit tricky to incorporate those statistics. So then we conducted the virtual 1,000 VE trials to estimate the sample size that gives the 90% or higher potential possibility of success for the VE. So then we came up with the N of 24 to conduct the virtual bioequivalence trial. So first we used the commercially available virtual bioequivalence. This was done with the SimSiv version 20, and at that time, version 20 virtual bioequivalence only gave us the 5 different GI physiologies to incorporate into the within subject variability. Because the tofacitinib is such a very well-behaved, high-soluble drug, none of those differences really didn't reflected on the within subject variability. So as you can see from the upper panel, the predicted variability was much smaller than actually observed. So this will show the bioequivalency between 2 different formations, but it looked very optimistic estimate because it's not predicting the observed within subject variability. So then we took another statistical approach to actually incorporate observed inter-subject variability. So we again simulated 5,000 populations using this simulation tool to populate the both reference and test formations. Then for period 2, we incorporated the within subject variability with the known distribution. Then we sampled 24 randomly from the pool of 5,000 population that we generated. And then we conducted 1,000 different trials to show the bioequivalency. So the regulatory experiences from this is that both FDA and European agencies seem to agree that this approach is reasonable. So it was accepted as a method to conduct the virtual BE. So lastly, I just want to touch on the pediatric extrapolation, and I don't have the time to talk about how we actually extrapolate it to the pediatric population, and that involves the ontogeny setting. And as I said before, the adult IL was really looked at the DDI studies to set the enzyme kinetics right, but this extrapolation was not only the age difference between adults and the pediatric population, but population that we are dealing with in the pediatric is the patient's population, so that there is another complexity of how the disease state changes the systemic component as well. So that was done nicely because we had enough sufficient phase 3 data from pediatric populations. So another component, the important part was that what's the exposure that we need to be efficacious in pediatric population. And that was done with this exposure response relationship that we developed for the adults, and we had a limited information for the pediatric exposure response relationship. But using the adult relationship, we comfortably extrapolated the pediatric efficacious concentration from that. So as you can see, all those modeling simulations that we used for tofacitinib pediatric prediction is a combination of both PBPK and PKPD, and also population PK as well. So just as a conclusion, tofacitinib PBPK models were developed using multiple different formulations, multiple doses, food effect, all those different physiological changes to just build the confidence in how the model is mechanistically describing the absorption of the tofacitinib, so that we will be comfortable extrapolating that to pediatric population. And secondly, as I introduced, more realistic bioequivalence analysis was conducted by incorporating the observed intra-subject variabilities from the clinic, just because this case we could not predict the within-subject variability with the simulation tool. Multiple modeling and simulation tools, as I said, PBPK, POP-PK, PKPD were utilized to assess the pediatric extrapolation. And lastly, adequate and well-planned clinical studies are very important to facilitate the pediatric development plans. Thank you.
Thank you very much, Dr. Sagawa. The panel must please come to the front. And our panel is going to be consisted of the speakers plus Dr. Vaithiyalingam from CIPLA, who is virtual online. Dr. Fang is my colleague from the Office of Clinical Pharmacology. She'll be up here, along with Dr. Fotaki from the University of Bath. Dr. Lukacova from Simulations Plus, and Dr. Patel from Certara. Okay, those were wonderful presentations. Let's go ahead and get started with a question here. The first question is, do you think the relative BA study with pediatric delivery vehicles such as soft food or baby formula conducted in adults can serve as the basis for pediatric product labeling? Can PBPK modeling be leveraged for this concern? So I wanted to make a comment, I guess, before we turn this over to the panelists about risk. And risk is obviously something very important to everyone in this room. But we're regulators and from industry, and there is a group that's missing here, and that group is clinicians and patients. And so I'd like to make a comment about risk, and this somewhat goes to a comment that was made earlier about the lack of information. And so this comes from basically 30 years in children's hospitals. Before I came to FDA, I was mostly in pediatric intensive care units. But there's the risk of not having information. When you're dealing with a critically ill child and you don't have information, what do? Well, you gotta make something up, using the best information you have. And so I think we have to incorporate that into our assessment of risk and how much risk there is in using the tools that we've talked about today for labeling. But let me turn it over to someone else on our panel, and maybe Dr. Fang would like to comment first about do you think PBPK can be leveraged to use for labeling?
Okay. So for new drugs, the use of PBPK to predict food effect is more based on the API properties. I mean, because I'm from the Office of Generic Drugs, so I want to touch upon from the generic drug aspect. So it is really a much harder question to use the PBPK model to predict the food effect, because on top of the API properties, you really need to determine whether the excipient can cause a different food effect in the pediatric population. So to me, that's a much harder question. So we are definitely interested in this area, and we would like to see the reliable PBPK model that can really predict the food effect based on the API properties as well as the formulation features for the pediatric products.
Other panelists, Dr. Lukacova?
I can only echo those comments and everything that has been discussed here already today. The PBPK models, and we've heard that from, I think, Taiko, who said that PBPK models are built from the beginning, right? Especially at innovator companies. You have the model, use the model to do the prediction. Right? I cannot say whether we are ready to use it for labeling, right? But that shouldn't stop anybody to do those predictions. That's how we are going to learn what else is still missing and build the confidence. So again, I don't really have an answer. Are we ready there for labeling? I would say probably not for all the cases, even though I've seen cases where it was used. But we do need to keep making those predictions, even if you go for the study. That's the only way we learn when we are going to be there.
Okay, other panelists?
May I?
Else like to comment? Okay. Let me ask one more question, and then we'll see if there's anything in the chat from our virtual audience. So let's take this a step further and talk about the youngest pediatric patients, say 3 months to 2 years or 3 months to 4 years of age. And so how confident are we for conducting PBPK absorption modeling in this, the youngest age group? And Dr. Sagawa, would you like to comment?
Sure. So the confidence is depending on what we are trying to answer, and that's the first thing. And so the question, we can be asking two different questions. One is if we can translate the bioequivalency that we found in adults can be translated into those young children, that's one. And another is, can we predict the exposure in those young children? To me, the latter is very difficult to predict exposures in 3-month-old, just because I think we just don't have enough data, the very solid data, and data we may have is so variable. So where the variability is coming from. So, but extrapolating the observed BE between two different formulations in adults, how can that translate into the 3 month can be doable, depend on how much mechanistically we understand the formulations that we can describe in the PBPK in adults.
Thank you. Dr. Liu, I got the impression you were a little bit negative in your presentation.
Yes, yeah. But by the way,
What do you really think?
say negative, actually, I will say I encourage people to try, and nobody or no one can stop us to try to do what if scenarios. I totally agree with Kazuko's comments. We have less confidence about it. We don't have enough data. As I shared in my presentation, we have the right model, and do we have the right data to parameterize the model? If we don't have enough, I encourage people to do that, but make cautious when we do the decision making. Yeah.
How about Dr. Patel? Sartara's got an answer for everything, right?
I'll try. So, yeah, I think everybody agrees that people have to attempt. I think at the moment we don't even have data where the model hasn't worked or has worked. So first is people have to try, even though Dr. Liu highlighted lots of gaps in the data, but what if we try where the data is available and see how the model is performing for different drugs, different formulation, or if not, it allows us to do sensitivity analysis to see, what are the most critical parameters. Like if you don't have an idea about gastric pH, can we do a sensitivity analysis? Just go to the worst case that, okay, if the pH is rather than 1, it's 5, what is going to be the outcome on the bioequivalence? So the PPPK models are there, even though we may not have all the accurate data, but it allows us to do the risk assessment, which is a very big step forward. So we are not trying to say we replace the studies, but this is a way forward where we try to attempt to do whatever we can based on what is available to us and try to understand where it's working, where it's not working, or at least to do the risk assessment. So I think it's probably a more positive tone that we have enough to start, maybe not to finish, but we wouldn't finish if we don't start.
Okay, other panelists? Okay, nobody want to vote?
Just maybe one comment more, for all this after that we discuss.
Is your mic on?
It's not working? I think it's working. Yeah, maybe it was. So one point that I wanted also to make is, especially when we're talking about the bridging of the formulation from the adults to the pediatrics, I think that's a big story. There is also, when we talk about the food aspects and how you extrapolate this from adults to pediatrics. From some studies that we have done in the past, it was in a clinical study. We could see that this could be, you need to think really carefully about the type of the dosing condition. So the reference meal is not always the best case to do your adult study and to extrapolate this to pediatrics.
Thank you. Anyone else?
May I?
Something, going back to the initial comment you made, there is not enough data. So if you don't do PBPK, what is there? Just a clinician making a wild guess, right? So I say it's not going to be available. People are probably not going to do a lot of bioequivalent studies in pediatric. Even I'm not aware if there are in such a young age group. So basically, rather than having nothing, even though there might be gaps in PBPK, it's definitely a lot more helpful to add some more context into the use if the clinician has to make a decision on use of the drug in younger age group.
Yeah, thank you. And the extension of that maybe is a much simpler model, which would be renal elimination, for example. So we've had this discussion in the Office of Clinical Pharmacology on, so we have really good data about renal failure and dosing of drugs in adults. Our ability to get that information in pediatric patients is pretty limited, but do we put something in labeling? And, we've had this discussion, and I think we're leaning toward trying to help clinicians by basically putting the best information and labeling that we can. Yes, in the back.
Line audience.
Okay, yes, go ahead.
No, we need, we have some question from our live
okay,
audience.
from the chat, okay. Or live audience, please go ahead.
Gilbert, I think we have a comment from Siva online, one of the panelists.
Oh, yes, I'm sorry, okay.
Thank you, Visha. So my point here is for
You're muted.
extrapolation from, am I audible, Visha?
I can hear you on my end.
Okay. A panelist, are you able to hear me?
We may need to increase the volume.
Pump up your volume here.
How about now? Is it
Yes.
audible?
Good. Yes, that's good.
Okay. So my point of view is like, it's more comes from the physiology of the patient, right? So if you are thinking about the physiology of patients who are 4 years and above, and when the PK modeling is based on the physiology, exact physiology of the patients 4 and above, extrapolating to the younger patients needs whole lot of understanding about their physiology, which are in the transition. So we should attempt, like the previous speakers mentioned, we should attempt that kind of a modeling. There is no doubt about it. But since the growth, since the transition of how the anatomy and physiology significantly differs, their absorption, their environment in the gut is so dynamic as their growth pattern is. So it becomes challenging to extrapolate the data. So unless we have certain studies to prove or disprove, it will be difficult to extrapolate from 4 and above to the little patients who are 2 and less. That's all. Thank you.
Okay, thank you. Dr. Jereb, I was going to be monitoring the online chat. Dr. Jereb, do you have gotten any questions from the virtual audience?
Yes, there are many questions, but maybe I would just select those related to this topic of this session. So there was this question to Dr. Sagawa, just a clarifying question. For the pediatric modified release formulation, was a bioequivalence study conducted in adults to show bioequivalence to oral suspension formulation? Or was it also demonstrated by virtual BI?
No, the virtual bioequivalence trial was conducted in adult healthy volunteers.
Another question, Dr. Jereb?
I hope that's good. Then another question was, I'm not sure to whom it's related, but it was a question regarding in vitro data. Was there, with this study, what were the experimental conditions, like the buffer volume, the strength, the pH considered or adjusted for pediatrics? So for any in vitro study for pediatrics.
It's a question to me.
Who is that question
it's
for?
It's not directed to a specific person.
So I will try to answer my case with the tofacitinib. So the in vitro dissolution for the tofacitinib ML dosage form was conducted in 900 mils of media, not biorelevant. And that is because, like I said, the first ML dosage form, osmotic delivery, we always developed that dissolution data conducted in the 900 mils of media, how it translates to the in vivo. We established with the multiple drugs, multiple durations, and so we have enough database to do this IVIVC. And this is why we didn't need to do a biorelevant dissolution for the dosage form that we developed.
Anyone else want to answer that? Go ahead.
I may comment. I mean, I work quite a lot in this area, so in general, I don't know. Probably the question was referred to your study, for the with the University of Birmingham, I guess, that there was like some pediatric media there and the conditions, so you may give some details. But in general, what we have done in my lab was that a few years ago, we tried a bit of like to develop more aspects of biorelevant media based on the physiological characteristics in children, in pediatrics, I have to say, and develop media that were relative to the adult one. So always just to understand a bit of like how these differences, for example, in bowel salts concentrations in the pediatrics compared to the adult ones would impact solubilization release. And in terms of dissolution studies, you know, we are more keen on using small scale dissolution studies, and we use different approaches, or with a mini paddle or USP4 or the microdis and so on.
Thank you. Dr. Fang?
Yeah, so I mean, I'm certainly not expert in the in vitro experimental design for the biorelevant dissolution testing, but from my understanding, the university actually tested a wide range of different conditions. For example, they tested different volume, like from somewhere from 500 ml to 200 ml, and they also tested different compositions. And the interesting part is actually then they incorporate those different biorelevant dissolution into the PBPK model and use the modeling to help them guide, okay, which condition is more biopredictive of the in vivo PK. And we actually published this paper. I quoted in my presentation slides. If I would refer the person to that publication to get more details in terms of the in vitro testing condition.
Yes, thank you. That's Dr. Batchelor's study, and we can put that in the chat if you'd like. Thank you, Rebeka. Let me ask another of my questions, and then we'll come back to you, okay? Can we leverage BCS classification for risk assessment of pediatric products? And as you saw, that was one of the questions that we had for that pediatric food assessment that we now have submitted to a journal. But at the end of it, we were wondering whether BCS or a pediatric BCS classification along with PBPK could really help us in the food effect. So, Dr. Fang?
Yeah, thank you, Gil, for that question. I mean, from my personal point of view, the answer is pretty straightforward. It's yes. I mean, we all recognize the age-related difference between the adults and the pediatrics in the absorption. So if we can really develop the pediatric BCS classification, we can certainly use that for risk assessment. But I guess the point really is like, do we really have enough data to really develop the pediatric classification system? I think that's a bigger challenge here.
And we did between 2011 and 2014, there was an interagency working group between NIH and FDA. And we actually did an assessment of all of the pediatric products that were available at that time, and along with a lot of details about them and BCS classification, and that's all published. It was published in 2014. There are several reports there that came out of the FDA and the NIH. That was the interagency working group that we had. So we can be happy to give you a reference to that if anybody's interested in that. Yes?
That was 10 years ago. Do you think it's time to try to reinitialize again? There must be much more information available now.
Much more information available now, that's for sure. And particularly when we talk about some of the oncology products, we've got so many new pediatric oncology studies that are going on. It's critical. There is going to be a formulations workshop study supported by FDA and PQRI, Product Quality Research Institute. It's going to be at the end of February, and we're going to be talking about, a lot of the formulation stuff plus PBPK applications, and so that'll be the end of February.
So, Gil, actually, we talk about this database. Quite a few years ago when I was still working with you many years ago. I guess my curiosity is, did anyone ever try to take advantage of this pediatric BCS classification and really showed the bigger improvement or better prediction? I mean, that part of the data, actually, I'm not aware of it. Maybe you have better knowledge.
Well, it was funny. I was reading back through that 2014 report we had, and one of the statements in there says, "Would a pediatric BCS classification help us more?" Not much came out since then. And one of my former colleagues at FDA, Varsha Mehta, was at University of Michigan before she came to FDA, and with Gordon Amadon, of course, very, very involved in BCS classification. They wrote a paper in 2020 on, pediatric BCS classification, and shouldn't we do that? So it's not happening very quickly. Yes.
Yeah, I think this is Siva Vaithiyalingam. I think there are three components for the BCS, right? I'm not much worried about the solubility component because the pediatric dose, single highest therapeutic dose is going to be much lower than the adult highest therapeutic dose. So I'm not worried about solubility. That characteristic will pass. I'm not even worried about the dissolution because it is formulation dependent and the dose is going to be even smaller. So the major concern would be finding out the permeability. In the absence of in vivo PK data, the permeability is demonstrated by using Caco2 cells. So since the pediatric patients are in the growth curve, finding a suitable substrate for their absorption mechanism via Caco2 cells or any other cells is going to be challenged. So out of these three components, solubility, dissolution, and permeability for defining what a molecule is a BCS 1, 2, 3, or 4, I think permeability is going to be the rate limiter. So if we crack that, then it is essentially the answer is yes. I agree with Dr. Fang when she said, can we leverage BCS classification for risk assessment of pediatric products? I think we have all tools. It is just that someone has to put the resources for the permeability component. Thank you.
Yes, another comment?
Yeah. May I just add one point here about this pediatric BCS? I think it's, as you say, this is something that is discussed quite a long time now. And we had tried a bit of mainly to see if what would happen in terms of the extension of a biowaiver, for a pediatric formulation, thinking about if you have a pediatric BCS or keeping your BCS of the adult one. And again, as someone would expect, there are cases that you would be at risk, especially when your solubility based on the dose of the drug, would be on the risk and change in classification. And then in that case, probably a biowaiver wouldn't be applicable if you think the different volumes. And we did this study for some amoxicillin, I think, and some other drugs that were just on the edge that you would change the solubility. So I think that mainly, this point of the PBCS and all this, there are cases that if you follow just the regulations that you have, like for the adult formulations, it's not very clear if you can get a biowaiver for the pediatrics if you consider the different dose volumes.
Okay, thank you. Question in the back?
Yeah, hi, Sunit Shukla, Boehringer Ingelheim. I have a question, not related to pediatrics per se, but maybe before that.
You asked your pediatric question at the end of session 1.
But it's about BCS3.
Now you don't want to ask pediatrics?
Okay, I'll ask it in a different way.
Okay.
So it's about BCS3 biowaiver. So there is a recent publication, I believe it's from FDA and industry partners, where they looked at about 150 ANDAs which were approved over last several years. And for the ones which were not Q1, Q2, the one which were not Q2, but the excipients were commonly used excipients, all those bioequivalent studies were found to be bioequivalent at the end of the day. So which means for those commonly used excipients for BCS class 3, there is a possibility of biowaiver. For practically all the applications which were submitted or reviewed, they were all bioequivalent, irrespective of the Q2 differences in the commonly used excipients. So my question is, in that context, and that can extrapolate to pediatric formulation too, if there is a change in the formulation for a BCS class 3, can we use PBPK alone to ask for a bio waiver? And it's open for both industry and regulators here.
May I answer that? Just can I
go
start
ahead.
the answer?
Sure, go ahead.
So, Harshil, what you have seen is only the passing data. What you have not seen is the failing data. So there may be a bioequivalent study where they have changed the composition of the excipients that resulted in a failure, which FDA will see it, but public will not see it. So you have not seen the failure data with the similar commonly used excipients. Thank you.
Okay.
Okay, thank you. Rebeka, we have 5 minutes, so you have one more question from the chat? Oh, I'm sorry, we had somebody in the back.
Yeah, I couldn't help it. So I agreed with the previous comment on the pediatric BCS that by and large, probably there's not a lot of difference for solubility, but I submit, I don't agree with that statement for neonates, because there's at least a lot of data that says neonates have reduced bile acid concentration, therefore absorption could be reduced. So the issue we have when we talk about pediatrics here, right? Pediatrics goes from neonates to 18-year-olds, right? So I think there needs to be some nuances in the pediatric BCS, perhaps, to me, would have most value for the neonates. That's just my sort of...
Yes, thank you for making that point. We've been talking about 3 months and up, but in fact, neonates are a total different ballgame. Even for diseases where we allow extrapolation in most of the pediatric population, we don't allow extrapolation in neonates. They're really a different animal.
Agree with the speaker. Thank you.
Rebeka, back to you.
Yeah. So one question, again, related more to the topic of this session. It's regards to the virtual bioequivalence study. How was the virtual, or how it should be, pediatric population selected in terms of demographics such as age or body weight? In addition, what is a consideration about conducting the virtual bioequivalence study using pediatric populations stratified by age group? So any thoughts to that?
So I'm just trying to clarify the question. The pediatric bioequivalence studies, I mean, we do not...
Virtual bioequivalence study in pediatric population.
Virtual.
The virtual.
Virtual?
Developing the virtual population, that was the question I believe I heard.
The virtual pediatric bioequivalence studies.
Yeah.
Oh, thank you.
Dr. Sagawa used that in her example of you developed a virtual pediatric population. So how did you choose that? How did you design that virtual population?
Well, my question would be, would you conduct a virtual bioequivalence trial in healthy pediatric, or would you want to see in the disease states? That would be my question, because it will be so much harder to do virtual BE in disease population in pediatric. And once again, Taiko's comments, when you call pediatric, yes, I think 6 to 18 is relatively easier. Anything below that will become difficult. So how would you, we have a different doses with the different body weights, and the youngest one is going to be a difficult one to even conduct, including the variability that you observed, not based on the variability in the physiological condition, but the dosing administration that mixed with the formula. It's not the bolus dose, it's just sucking through for 30 minutes mixed with the pudding, then you are just refusing to eat over 30 minutes. All those variability, how do you consider those? It's not going to be a clean adult type of study to me if we do conduct such studies.
I see Dr. Rostami at the back. We have 2 minutes.
I'm not sure I can go for 2 hours on this particular issue. I disagree, with all the panel members that they mentioned that we haven't got enough data, I don't know, below 2 years old, etc., etc. I think this goes back to, to the philosophy again. Yes, we are trying here to actually look at, sort of the different drugs and then come up with a classification, something simple that everybody understands, sort of a class 1, 2, 3, 4, everybody can remember, even if they are 2 years old. But, the thing is that when we look at, for instance, the pieces that we have focused on the stomach emptying, with that philosophy, we use PubPK for gathering all the data that different people have published and put, the typical thing that every PubPK person knows, covariates with regard to what is determining these elements. We were not focusing on any drug. This is again the same with every other aspect of the GI tract. Believe me, there are loads of data already in the literature, but you need to focus and actually look at them with a different philosophy, gathering them, analyzing them for the purpose of, going back to what actually Kazuko was talking about, that, whether the differences are the patient, sort of the disease state, whether it is, of course, if they are healthy, they are not going to go through some of these, studies for physiology. This is, in itself, is difficult. Most of these physiological studies, in fact, are done in patients, and that is, I would argue, it is the better one, because that's what we need for these. So I think we haven't done enough because of this wrong philosophy of focusing on the drug. Those drug data, whenever available, even they are sparse, etc., together can help us to do some verifications. But we should not expect to see, on the basis of focus on drug, to come up with patterns that they are easy. University of Leiden did that. You know better than me, actually, Gail, that they showed that even for a simple case of 3A4 substrates, they couldn't come up with a pattern. And I think, again, it goes back to philosophy. Otherwise, the data for physiology, yes, there are gaps, but many of those actually fundamental physiological, biological values are available, but sparse and scattered, and we can use AI and all the other things to gather that data.
Well, Dr. Rostami, you had the last word. So that brings this session to a close, and now it is up to Dr. Lionberger to wrap it up. Thank you very much, panel members.
Thank you all. So this is the closing for the virtual part of our workshop. We will continue with some related to the 3 sessions today for some more and interaction, and then we'll come back with some summaries for the whole group. For those of you online, we'll be releasing the summaries of those discussions through various ways. We have workshop reports as well, so to the learnings that come out from those. I encourage you to think about, as we have these workshops in the future, to attend in person and gain some of these valuable in-person interactions that we have along with the virtual content. So again, the title of this workshop is Regulatory Utility of PBK Modeling. And I want to have a discussion around that, because I think even before the regulatory utility, the important thing is to think about the utility of the PBK models, right? The utility is not because FDA says submit it to me. The utility of these models is how valuable they are to your development programs. Let me give examples. Just even inside FDA, we use these models extensively for various questions that we have to answer. Oh, we're developing a guidance. What study should we recommend? Right? We use the PBK models extensively in analyzing those questions. It doesn't mean that then, oh, well, we use it to develop a guidance. You have to submit a PBK study to use that result. So there's huge value that's generated from PBK models, even if you don't submit them to FDA. And I encourage, one of the things for the generic industry, especially to learn from this type of workshop, is to really recognize the value that creates. And that's why, we've invited speakers from across different parts of the drug development industry, so you can hear that perspective. But I think that's the important part of the message here is these are extremely valuable across the development. As you're planning a BE study, as you're making formulation decisions, what should my formulation do? How fast should my dissolution be? What should my dissolution be for the different strengths of the products I'm developing? All of these type of questions that are common to every solid oral product generic development question, there's utility in the PBK modeling and absorption modeling approaches for input into those. as well as setting specifications on dissolution, setting specifications on particle size, all of these things, extremely valuable to you, even if you don't submit them to FDA. And as I said, the utility of the models has to come first. One comment struck me today in the meeting was someone said, well, I tried PBK modeling once, I sent my model to the FDA and they didn't like it. Therefore, PBK modeling has no regulatory utility. And I think that idea can't be the way that you think about this, right? The idea is the utility internally has to come first. But there are situations where when you want to do, it's not just helping you, the models help you meet FDA's expectations, but there are places that you will want to share the model with FDA and use that as a frame for discussion. But that's a smaller set than the places where the PBK model has utility. But if you don't build that foundation and you just say, the first thing I'm going to do as my company gets into PBK modeling is I'm going to send this model to FDA. I don't think that that's a pathway for success. It's built on the foundation of using these tools effectively internally for all the decisions that you have to make in drug development. And then I think you'll be much more successful when you want to do more challenging regulatory situations where discussion of the model with FDA is going to be really important. And I have a few comments on that. One of the things that we've seen is, here's a PBK model instead of a BE study. And it may be a PBK model that describes some results. But the challenge there is that model really considering the factors that are important to the study that you don't want to do? So for example, if you say, I have a formulation difference and I don't want to do a Fed-BE study, I have a PBK model of the drug. That's not going to be compelling to FDA unless you have a PBK model of the change that's relevant to the change in formulation or understanding that effect. So you have to think about the purpose of them before you set to FDA. And I think that's something that, as we talk about in workshops like this, really focusing on what are those questions in the pediatric space that we just talked about, in the food effect space, especially there, it's really, how do we get to the models to the point where they actually answer the question about the change that I'm looking to make. And then they'll be even more useful in those cases and specific cases. And, another challenging place is, my study failed. Now I'm going to do the PBK model and I'm going to send it to FDA to try to justify the failed study. Again, this is not a pathway that leads often to success. And that's why I recommend, if your approach is the modeling and simulation is always built up, then when you do encounter a failed study, it's an opportunity then to integrate that into an improvement into the model, right? So if you think about, you've heard a lot of examples here about, use a fast, medium, and slow formulation to describe that, validate the model. Right, if you end up with a bunch of failed formulations, you might say, I wish I had a modeling approach from the beginning where I would have learned from each of those 3 failures and then had a model that FDA would believe, and the 4th study I'm going to pass because now my model works and I can use the model-based approach to say, here's the formulation that I actually want to do. I don't have to repeat a study that might be within the space of those failed studies, even though my 4th formulation, the one I'm going to submit, might not be the same as the other 3. You have then the opportunity to submit that directly without a new BE study. If you have the perspective of, I have a plan, a system for how I build up the knowledge internally. So that's why I said there's a great value in the utility of these approaches and thinking about what a sort of modern drug development program looks like for the generic industry. And I think that's an important part of this. And I want to say, just from FDA's perspective, we have some new ways that we're thinking about to help when you are bringing a model to FDA to improve the chances of success in that. So one is, as was mentioned several times, our model-informed evidence pilot program. So this is a new meeting type that you can use. And want to be clear what the scope of this is when we talk about the model-informed evidence. It's a kind of approach where you might be using a model along with some other type of in vivo study to put that together into a holistic conclusion about bioequivalence. And so this might be a more complicated, for complex products, might be a more complicated situation. Again, it's not sort of just like the model solves everything, but it's integrated into how do I use the data that I collect, build that into a model that might then be useful in other situations. But this is a type of meeting where you can get feedback on the model and its validation before you come into the regulatory question. So I encourage you to look at that program. And in cases where that's part of your planning and thinking to, use that pilot program, provide information. The more successful this pilot program is, then we can do more of these in the future if there's sort of a sense that this is value and it creates value for you. But it's really focusing on getting that early feedback so you're going to be successful in the PBK modeling. Another aspect that we'll be hearing about, and it was mentioned, we have a workshop next year on the model master file approach. So again, as the PBK models become more useful and more gets submitted to FDA, right, we have to manage effectively the review and evaluation of this. I think in a lot of the modeling approaches, and apologies to my friends in the Office of Clinical Pharmacology, a sort of a small number of applications, each model can be reviewed independently. You think about the generic drug program, scale of 1,000 applications a year. We want to have efficient ways where we're not reduplicating evaluation of different models. And the model master file approach is a way to, for FDA to segregate the review of the model from the review of the application. And so if a model is used by multiple applications, it can be reviewed once. And then just the reviewers of that application can say, is this appropriate for this purpose? But I'm not repeating things that other parts of other people in FDA have already reviewed. So there's lots of opportunities there for reusability, for FDA efficiency reviews, but also if you think about experiences with drug master files and knowing that your supplier of a model has a model that FDA has already evaluated, that gives you more confidence that this approach is valuable. So it can be useful as well for the environment and the ecosystem of modeling as well to do that. So we'll be having a workshop on that concept. But again, this is, ways to make the incorporation of models into the regulatory submissions more effective. But that's not the, again, as I said at the beginning, that's not the main point. The main point here is the utility of these systems internally within your own organization in effectively using them to make your development programs more effective. That's the foundation for the successful use of these models in your regulatory submission. And so with that, I think that's the end of our virtual component of this, and we'll continue here with our in-person components. Thank you all very much.